This is table 2, and this is our presentation on mitosis and meiosis. This is Michelle, Ying, and Dung, and I'm Oscar. We're going to start off with mitosis first, which is a cell division from one parent diploid cell into two daughter diploid cells.
So in interphase, the chromosomes are not condensed yet. Also, they are enclosed in a nuclear envelope. Moving on to prophase, the centriole begin to appear and sister chromatids have been duplicated, and the nuclear envelope began to disappear. In metaphase, the sister chromatid line up in the middle of the cell, and the centriole move to the side and attach itself to the kinetochore of the sister chromatid. In anaphase, the sister chromatids are separated, and the kinetochore are disappear from the sister chromatid. In telophase, the centriole move further away the sister chromatids away from each other, and the cell begins to have a cleavage plural, which is an animal cell. In cytokinesis, nuclear envelope begins to form again, and the cell produces two identical daughter cells to the parent.
Now we're going to move on to meiosis. Meiosis is similar to mitosis, only that meiosis takes one parent diploid cell and turns that into four daughter haploid cells.
So in meiosis, like mitosis, interphase also occurs. However, take note, the interphase is not part of meiosis. It is just a process before meiosis that occurs. So in interphase, the nuclear envelope is not yet dissolved, and the chromatins are still tangled up inside the nucleus. When we move into meiosis in prophase 1, the nuclear membrane begins to dissolve, and crossing over happens at the chiasma. A chiasma is the location of where crossing over occurs. Now moving on to metaphase, the spindle fibers begin to attach themselves to the homologous chromosomes at the centrioles, and the homologous chromosomes line up at the metaphase plate. And when moving on to anaphase, the spindle fibers pull apart the homologous chromosomes, and that leads to telophase. Within telophase, the cleavage furrow appears, and the two cells begin to separate.
Then we begin the second division of meiosis. In prophase 2 of meiosis, there is not much happening in there, and it pretty much stays the same. So moving on to metaphase 2, the chromosomes line up in the middle at the metaphase plate, and then at anaphase 2, the sister chromatids are pulled apart to opposite sides of the cell. In telophase 2, the cleavage furrow starts forming between the two halves, and then at the end of the process, there are four daughter cells.
Now we move on to comparing and contrasting both mitosis and meiosis. One main difference is that in mitosis, you get two diploid cells, which is a cell with 46 chromosomes, while in meiosis, you get four haploid cells, which are cells with 23 chromosomes. In mitosis, the daughter cells are identical to the parents, but while in meiosis, each of the daughter cells are unique to each other and not identical to the parent due to the cell crossing over. In mitosis, there is no crossing over, while in meiosis it crosses over during prophase 1, which is the chromosomes sharing DNA with each other. A similarity that they both have are that they're both types of cell division. They both separate into 2 or 4 different types of cells. They both have a nuclear envelope that dissolves in prophase. And lastly, they both have cytokinesis, which is a cleavage furrow in the animal cell, while in plant cells they have a cell plate. And thank you, and this is our presentation.